Hello boys and girls, this is Mr. So. Welcome back for another lesson of biology. Today we are going through lesson 27 of chapter 27, Protein Synthesis, the classwork 27.1. Before you start watching the video, remember to like and subscribe the channel and also share the video to your classmates. Let's start. Let's do a question in your classwork 27.1. The following diagram shows the process of protein synthesis. And as you can see, you can be able to see the ribosome, the tRNA, the mRNA over here. So this is the process of translation. All right, so question A. Name the structure 1, 2, and 3. Let's take a look at structure 1. Structure 1, you can see it over here. It is carrying an amino acid. If it is an amino acid carrier, it must be the tRNA, transfer RNA. You can just write it short as tRNA. What about structure 2? Structure 2, you can see it has the codon. If it has the codon and it is a straight chain, it must be the messenger RNA, the mRNA. And for structure 3, it is a ball-shaped structure which holds the mRNA and also holds the tRNA. This one, structure 3, is the ribosome. And then let's go to question B. Can you state the function performed by structure 1? Structure 1 is the tRNA. The function is it is an amino acid carrier. And the function is to carry the amino acid to the ribosome. Let's write down. tRNA brings or carries amino acid to the ribosome. And then add it to the growing polypeptide. And all together in here, you get 1 mark. It's a little bit difficult. You have to write a lot. Describe how it carries the amino acid, and after it carries to the ribosome, what is the function? To add the amino acid to the growing polypeptide. All right, carries amino acid to the ribosome. Question C1. Can you name the coding unit X and Y? Where do you find the coding unit X? You find it right below the tRNA. Well, for the coding unit Y, you can find it right on the mRNA. So what is the coding unit X and Y? X, it should be on the tRNA, it should be the anticodon. Well, for Y, it's on the mRNA, it should be the codon. And what is the relationship between them? The anticodon and codon, they have a complementary base pair manner. And then question 3, what is the importance of having the complementary base pair manner? This is to ensure a very correct matching between the anticodon and codon. And it also ensures The sequence of genetic information in the DNA can be transmitted to the translation process. Ensures the sequence of genetic information in DNA can be transmitted to the translation process. So altogether in here you have 2 marks. Correct matching and ensure the information, the triplet code in the DNA can be transmitted to the translation process. D1, according to the base sequence of structure 2, that is the mRNA, can you state the DNA sequence on the template strand? So how to do the questions? Let's draw out a very simple diagram of DNA again. Draw it out. And then because you know that template strand is on the lower part, the above is the coding strand. And then the template strand is complementary to the mRNA. Alright, so one of the methods to do the question is you write out the sequence of mRNA and then write the complementary base pair of the template like this. So for example, you have AUG, ACG, and GAU. So now you can write out the template sequence, the triplet code as T, TAC, TGC, and CTA. So this is one of the methods to get the answer. However, you have an even better and faster method to tackle this question. That is, as you know, the mRNA is complementary to the tRNA. This is the mRNA. And structure 1 is the tRNA. As the tRNA is complementary to the mRNA, you can think about the tRNA sequence is actually exactly the same as the template. But the U base in the tRNA is changed to T base in the template. So you can just copy the tRNA strand but change all the base U to base T. Then you will also get the answer like this. So you have copy it down, UAC, UGC, and then CUA. Now, because in the template strand, you are actually a DNA molecule. You don't have the letter U, you don't have base U, so you change the base U to base T. Change the base U to base T. So now, do you get the same answer? Yes, you do. So you also get TAC, TGC, CTA in the template strand. And according to the genetic code, can you write down the amino acid sequence of the polypeptide synthesized from the above segment of structure 2? So let's take a look at this table. This is a codon table. And you can find the codons on the mRNA in this part. So what you need to do now is take a look at the codon and then match it one by one with the amino acid. So let's start with AUG and then ACG and then GAU. Let's look for the amino acid, which is coded by the codon AUG. It should be the first one, AUG, as here, methylene, MET as the start codon. And then for the second amino acid, it is coded by ACG. Let's look for ACG in the codon table. ACG, it is found in here as the THR, this amino acid. And then the last one, GAU, we can find this codon coding for the amino acid as ASP. So now you get the amino acid sequence as MET, THR, and ASP, 3 different amino acids joining together to form a polypeptide. For the very last question of this classwork, can you write down the last codon of mRNA given? And what is the significance 重要性 of this codon? So for the very last codon in the mRNA, it should stop the protein synthesis. It should stop the amino acid joining. It should be a stop codon. So the stop codon is from the table. You can find it as here. You have 3 different stop codons. So let's write down. It can be UAA, UAG, or UGA. All of them, they are the stop codon. So, and the significance is they are stop codon which ends the synthesis of polypeptide. That means it ends the joining of the amino acid. You get 1 mark in here. And then after you finish the synthesis of polypeptide, it allows the polypeptide to release from the ribosome. The completed polypeptide can be released from the ribosome. Okay, you get another mark. Altogether, you get 3 marks in this question. Last but not least, can you go to page 9 of your textbook to do the MCQ? Also go to page 20 of your textbook to do the MCQ question 4. That's it for the part about the protein synthesis translation. I hope you enjoyed the video. I will see you in the next lesson. Bye bye.